All right, here we go. This is a new lecture. Today we're going to discuss this paper. Oh, wait, I have to record multiple audios in case something bad happens. Okay, hello. Today we're going to discuss this lecture. A rationally enhanced red fluorescent protein expands the utility of FRET biosensors. So this topic is on protein engineering fluorescent proteins. A quick comment that I said in the Discord. I completed reading analysis of this paper. Read what makes sense. Don't fret over things that don't make sense. So some of this paper was a little bit hard even for me to understand. I understood probably about half of it, 50%. And when you are reading a paper, a primary literature article, it's kind of like let's see, if you were to graph like input of energy, like in terms of like actually trying to understand things and amount, quantity learned. When you're reading papers, it's kind of like this, like a minimal input to trying to figure out understanding things will help you learn a lot. And then after that, there's kind of like diminishing returns of you trying to understand everything. So when you're reading a scientific paper, just try to gather and glean as much as you can. Try to dissect a few things that you feel like you might be able to understand. You're on the cusp of understanding, but then don't spend 8 to 12 hours, trying to understand everything. So just understand that it's kind of like diminishing returns while you're reading papers. And I just say that because this is the first paper that we have actually discussed. Okay, so let's just first think about the title. Just looking at the title of a paper should tell you what it's about. So tell me what you guys think. What's your interpretation of, like, what's this paper about? Yes. So they're developing biosensors. We'll talk about what that means. They're working with red fluorescent proteins, so trying to make them essentially more bright. What does it mean, rationally enhanced? What does that mean? Yeah, like they thought about it. They strategically thought about, well, what mutations might help this thing? And then they explored those as hypotheses to try to engineer the protein. Okay, so we understand the title. Let's go into some basics of fluorescent proteins. So we've talked about fluorescent proteins many times in this class in the past. There's GFPs, greens, RFPs, reds, CFPs, which is cyan, blue, YFPs, yellows. There's all kinds of different colors. And the brightness can be engineered. So when they first started, if you read the introduction, they kind of talked about 4 different variants of red proteins that come up in this paper. So just to quick help you guys understand, they talked about M cherry, tag RFP T, and stag RFP T. So M cherry was they kind of said like this is one of the more weak ones. And then they talked about this one they're particularly interested in from coral, tag RFP. And then tag RFP is a derivative of, or tag RFP T is a derivative of tag RFP. What was the difference between this one and this? Did anybody catch that? They did eventually do that, but that's not the main thing that causes this to be different from that. We'll talk about that in a second. Yeah, tag. Well, no. Okay, so tag RFP is the wild type, and then what's the difference between this one and this one? Just looking at the names, what would you think? No, this one was a mutation. So this was actually like some kind of mutation annotation, where this was a single. If I don't, again, maybe you didn't catch it, but this was a single point mutant derivative of tag RFP. So they took the original wild type, they mutated one residue, and just from one residue, they were able to double the amount of fluorescence. That's what they had said. Okay, so they're already starting this project working with sort of like an already once fold enhanced RFP. And then throughout this paper, they start talking about this one. Did anybody catch what the stag RFP was or what this stands for? Yes. So this is like that's what the S stands for, is super tag RFPT. So this one then is a derivative of the already enhanced one. So now this one is double enhanced, which is why they call it super, the stag RPT. So what you'll kind of learn from this is everybody, when they write papers, they kind of have their own naming systems, and they're kind of allowed the freedom to name the proteins that they are, I guess, developing. And so some of these are wild, some of these are wacky, but there is some logic, I guess, in this paper to how they're naming these things. And essentially, these are all brighter variants in ascending order. Okay, in the introduction, they talk about some of the cons of this coral fluorescent protein. And they talk about this concept called dark state conversion. Did anybody understand what that was? Or can you help me understand what that was? Did anybody catch that? Yeah, so you caught the basic gist. Essentially, what is happening is this tag RFP protein has some conformation that is generating fluorescence, but there is a dark state of it where it can go through a conformational change into some different morphology, some different tertiary structure, and this one is no longer fluorescent. So one of the cons they talked about is that this particular protein is kind of like blinking. It blinks, and it goes through this on state and this off state, and that all has to do with the conformation of the protein. And there is this chaperone, which is a protein that binds to it, which mediates this switch. So this protein called DRONPA, I've never studied this. Like, I don't know a ton about this. Again, I'm just learning all this from the introduction. There is this other intermediate protein called DRONPA that when it binds to the tag RFP, it will cause it to go through this conformational process into the dark state. Okay, so there's kind of like a problem. This is one of the cons, is that there is this sort of like weird chaperone that shifts it from dark state or from light state to dark state. Okay, and so they essentially see that as a problem, and they want to engineer this in a way where that process no longer happens. Okay, so that is sort of like the gist of what's happening in the paper. Let's see, before we talk about that, we'll talk about FRET. So now just quick intro into FRET, and I am not an expert on FRET. Like when you start to get into molecular biology, you'll realize that everyone sort of has their own methodological expertise. So I'm not an expert on FRET, but I can understand what they're doing. So essentially what FRET is you can take two fluorophores, and the classic example is CFP and YFP. So if you take a CFP, let's say this is CFP. I'll zoom in on this. CFP and then YFP is yellow. Let me get a yellow here. CFP and YFP. Okay. What FRET is when you excite one fluorophore. So the way that fluorophores work is they all have some what's called absorbance spectrum, where they take in light energy at some absorbance wavelength. And so that's like essentially like exciting the fluorophore. And then once it gets excited, it will fluoresce and expel photonic energy, photons, at a certain wavelength. That's what causes it to shine light. And a FRET reaction is when one fluorophore shines, it takes in energy and then expels the energy. And the energy that's expelled is in the exact wavelength that excites the other fluorophore. So in this case, if the FRET is happening, CFP is taking in the energy, it's fluorescing, and it's exciting the other fluorophore. So if this happens, what you see is then yellow fluorescing, because the blue is actually exciting the yellow protein. Okay, and the reason that this is useful is because this phenomena, and FRET stands for fluorescence resonance energy transfer. So it's transferring essentially the fluorescence. The reason that this is useful is because you can use this to measure distances between proteins. And so it's a useful tool to test whether or not two proteins interact. Because when two proteins interact, they'll come in close proximity. And then that causes this FRET phenomena to happen. Okay, so the way that you measure whether two proteins interact is you can create tags of these constructs. So imagine the CFP, you create a fusion to some protein A, okay? And you, on the YFP, you create a fusion to some protein B, okay? And if these two proteins interact, they will come together and they'll bind together, and that brings the fluorophores together, which then causes the FRET phenomena to happen. Okay, so again, it's a way to measure whether two things interact by creating fusion constructs. Are there any questions about that? Okay, so we all understand sort of like the basic gist of FRET now, and its purpose. Okay, so then what would you say was the goal of this paper? Yes. So that's a very good way to say it. Increase the efficiency of FRET with red and green systems. So when this paper was coming out, most of the FRET systems are CFP, YFP, where those are the ones that work in the FRET system. But there are other reasons you might want to use different fluorophores. Maybe you're working with other stains, or maybe you have some particular cell system that really works better with our reds and greens. So they wanted to engineer a different FRET system that is essentially a red marker shining and or actually a red and green system, where a green marker is shining and causing FRET transfer to the red fluorophore. Okay, so that is essentially the goal of this paper, which is then going to require like protein engineering. So I said they want to build a GFP RFP FRET by re-engineering these proteins. That's exactly what you said. Very good. Okay, so hypothesis one. Actually, before I just showed the answer, but like, what was in the title? It says it's rationally enhanced. So what was their rationale, which is kind of right here? But did people catch that? Can you explain that to me, their rationale? Yes. Okay, so this was the protein that they're working with. This is the red, the RFP that they're working with. Okay, and the way that these molecules work is there is essentially like a barrel structure of beta sheets that wrap kind of the fluorophore. And their hypothesis is that the reason that this protein is blinking is because there are residues on the outside of this protein. Here is one that they highlight, D159. There are these acidic residues. So these are negatively charged amino acids that are important for the interaction with that chaperone. And the chaperone's name was Drampa. Okay, so their hypothesis is that these charged residues on the outside of the protein are actually like an interface through which this Drampa protein binds to this protein. And if that binding is allowed, that's what causes it to transition to the dark state. Okay, so their hypothesis is if they can mutate the protein in a way that inhibits that interaction, they will remove the ability of Drampa to bind, and that will create brighter fluorescence. Okay, so that's essentially like the hypothesis of what they're working with. They want to mutate some of those residues and see if they can reduce the interaction of Drampa to increase the fluorescence. And so they create this system where they actually fuse the RFP protein to this Drampa. Okay, so in here, in this case, Drampa is this green thing. And when it's bound, it's in the negative off state. And so they created, again, like a fusion of this RFP fused to the Drampa. And the reason they create the fusion is because they sort of want to be able to know whether or not they can successfully inhibit that binding. Okay, so you can imagine if it's binding, it will look, the linkage would look something like this, and this would be off. And if it's not able to bind, the linkage would look something like this, and this state would be on. And so their hypothesis is, can we make functional mutations in this interface that essentially inhibit the ability of these two things to bind and cause this thing to fluoresce permanently and more brighter? Okay, that's the gist. Okay, let's link. Okay, so then the next question is, okay, so you know they're going to do the mutagenesis. And they say here, I took this quote, we performed random mutagenesis at 4 solvent-exposed charged residues on the tag RFPT fused to the Drampa. So they start doing the mutagenesis. And the next question is, how did they do the mutagenesis? Did anybody look into the methods, and were you able to use your biotechnological knowledge to discern how they actually did the mutations? Here's where you guys contribute. So, okay, well, what does random, okay, so yes, let's talk about random mutagenesis. That is part of the answer. Random mutagenesis, what does that mean? So there's four that they're targeting. So they're not randomly mutating the RFP anywhere haphazardly. They're targeting four sites and they're using random mutagenesis. So what does that mean? What do you think that means? Yeah. So essentially, they're taking these four sites. Actually, the better term for this, it's not called random mutagenesis. This is what's actually called saturation mutagenesis. So essentially, they are generating clones where at those four positions, there is every possible amino acid. So essentially, they're generating a whole bunch of mutants where at each of those four positions, you can have the option of having all 20 different amino acids. So now that you understand this, so that's what saturation mutagenesis means. It means is you're testing all possible possibilities at one position. In this case, they're testing all possibilities at four positions. So now that you understand that that's sort of like the strategy, what is the mechanism or how do they actually do that using like biotechnological methods? How do they do that? So they do express these in E. coli, like they do tests in E. coli. What how describe to me how those tests were done? Let's think through like the process of what needs to happen. What what needs to happen? And here maybe I'll just show the relevant paragraph. Okay, so here's one of the paragraphs that deals with like how they do this. Okay, so keywords are libraries of single and multiple site mutations were generated. So they're generating libraries. Okay, so again, libraries are many plasmids. So whole cultures of many plasmids. So you're going to have to generate some mixture of a whole bunch of different plasmids where in that site, I'm drawing it blue here, but it's actually RFP. In those positions are different mutants. Okay, how do you generate those? So they are going to use site-directed mutagenesis. So we know how that works. Like we've talked about that. But the interesting thing is, again, so when I taught you guys site-directed mutagenesis, I taught you how to make one very specific mutation. Like here's how you change residue from A to B. That's not what they're doing. They want to change residue A to all the gamut of all 20 at four different sites. So looking at this paragraph, the key word here is degenerate primers. How would you modify the site-directed mutagenesis to do saturation mutagenesis? Yeah, so essentially they're running like one reaction, one site-directed mutagenesis reaction, but they have a mixture of primers, which they're calling degenerate. And what it means is they're degenerate at that position. So wherever they're doing their like mutation, again, imagine what's happening here is they have their quick change primers, and the quick change primers are PCRing around the plasmid. Except they have, instead of one primer set, they have a suite of primers where at that one position is all the possible different codons. And the reason it's called degenerate is because it's essentially like you don't know what the sequence is going to be. It's just like all options are possible with these degenerate primers. Okay, so from that reaction, you can see how this reaction would then generate not just one mutant, but all 20 different mutants at that position. Then they transform that plasmid into E. coli, and that then is the library. The clones recovered is that library. And how do they figure out which clones they were interested in? So imagine they do this site-directed mutagenesis reaction. And again, let's see what's happening here. So they have PCR'd around the plasmid. They have caused some mutation, which is degenerate. So at this position, you have all 20 possibilities within a population of plasmids. And they transform that population into E. coli, and they put it on plates. And what do they see on the plates, and how do they know which clones they're interested in or not? Yes, and that's what they did, and that's what they say they did. They just looked at the colonies, and amongst the colonies of these transformed population of plasmids, some of these would be brighter red, and some of these would be weaker red. And so they're able to pick the colonies that they're most interested in, that are the most bright red, quite easily. You pick these colonies, and then you sequence them, and then you figure out what it is after the reaction. So it's kind of like you're introducing randomness, stochastic randomness, but you have a phenotype that you're selecting for. And then you pick out the clones that have given you a phenotype that you want, you sequence them, you figure out what they are. Okay, so they did this at 4 positions. And what was the discovery? Yes, there was only one that stood out. Very well. Let me see if I can fix this. It's still recording, apparently. Okay, so the one that they chose is D159V. That was the one that gave them the brightest fluorescence. Sorry, that caused me to lose my train of thought. So let's try to understand what's happening in this figure. So look at this data. This data is actually pretty easy to understand. So first off, is this measuring a colony of bacteria, or what is this actually measuring? What's the thing that's being measured? Yeah. Yeah, so this is measuring essentially absorption and fluorescence. That's the y-axis. So this is essentially like how much light it sucks in, and this is how much light it spits out. But my question is, what are they actually measuring? Is this like a colony of bacteria, or what is this? This is actually the protein. So once they found the colonies that they were interested in, the one colony, the one mutant that was pertinent, then they actually did protein expression. So they actually created a his-tagged variant, and they did recombinant protein expression. They expressed the protein, they purified it, and that's actually what this is. So you're seeing here an SDS page of their actual purification of this mutant. So here's its purification. What would you say about the purification? Is this a good purification or a bad purification? It's good. It's really pure. Although, what would be one criticism of this gel? How do you know it's pure? There's only one band, but you know it's pure up here, but what has been left out? They cropped the rest of the gel. So this is actually kind of like one shady thing they did, is this looks pretty pure, but there might be some garbage down here that they did not want you to see, and they kind of cropped that out. We'll give them the benefit of doubt, but that's sort of like, you typically don't want to do that. If you're trying to show purity, you really want to show like the full gel to show how pure it is. Okay. But okay, we'll take their word for it. They're expressing this protein, they're purifying it. Then they subject the purified protein to essentially like a fluorescence assay where they're shining the light in. That's the absorbance. And you can measure how much things absorb. So compared to this is the first version of the engineered RFP, and this is the super version of the engineered protein. You can see that it's absorbing more light. Okay, so the pertinent data is right here. It's absorbing more fluorescence, and this is the absorbance wavelength right here. Okay, and then this is actually the fluorescence. So here's where it's spitting out energy at this wavelength, and you can see that the amount of fluorescence that it's spitting out is much higher than the other engineered variant. So they're successfully able to demonstrate that they can actually engineer the protein to get brighter via this saturation mutagenesis protocol. Okay, are there any questions about that? Okay, I need to think about it next. Yeah. In theory, I mean, this isn't a pretty good publication. When I submit to this journal, and if I put a gel like this, they'll ask me for the raw image. So you usually will have to show the raw image to demonstrate that you're not doing something shady. On the other hand, it depends what the reviewers say. If the reviewers don't care or they don't raise that, then you'll probably like slip through. So it all depends on what the reviewers think. What happens if you get caught? You'll get in, if you're actually like manipulating data, like you'll never be in science ever again. So like strict warning here, never ever do that. If you're doing something that's sort of like in this case, I don't know, like if they're purposely trying to hide something, I doubt that they're like trying to hide something, because this paper is kind of like a simple paper. But again, you always want to err on the side of safety. You never ever want to be accused of doing something shady like that. And I'm not, just to be clear, like I am not, because I'm posting this, I am not accusing these people of doing anything shady. I'm not doing that. I'm just saying that in general, you will probably want to show more of the gel if your argument is to try to show the purity. Okay. Then the next thing that they start trying to address is, okay, so they have this engineered, very bright fluorescent protein. Question 2, can this protein be used in FRET? Period. Okay, so here's where things start to get pretty complicated. And you start to look at this figure, and this is kind of like the main figure that you need to try to decode in order to understand what's going on. So this was the hardest part to decode. And I can help walk you through that, but I'm curious to see how far you guys got in this analysis. What is being documented here? First of all, what are these things? If you imagine this as like a y-axis, what are these different entries? Okay, so these are different version constructs. Okay, but it's not just the RFP here. So the RFP is right here. The section of the fusion construct that's dealing with RFP is here. And then what section is this? What's in this green section? No, they're actually not doing that here. They're not testing whether A is interacting with B. What are they actually doing? Like if we were to draw out, so you have some kind of a construct that has an RFP fluorophore here, and it's linked to what's over here. What are these? What is this? Yeah, this is green. This is green fluorescent protein. So these are different versions of greens, and these are different versions of reds. So you have scarlet, M cherry, stag RFP, M ruby, and here you have different greens. Clover's a different green. EGFP is enhanced GFP. So they're testing essentially like different combinations. They're creating this fusion construct where by default, these should always be FRETting because they're fused close to each other. So just imagine that when you build this construct, you're fusing these things close together so that the FRET is in a perpetual state of like exciting each other. That's essentially what they're trying to do. And so they have different versions of green and different versions of red. Why would you want to test different versions? To see what causes the best FRET combination. Because remember what has to happen in FRET is one of these has to get excited and it has to pass the energy to the other one, which then gets excited. And all these are passing out different approximate wavelengths. And so one of these might work better. So that's actually what they're measuring. Okay. Then there's a couple things that are worth explaining. So what is a linker? What is this? A linker that is falling somewhere in between these things in some of these cases. Yeah, it's random. It's probably not random. Like it probably has a very precise sequence. It might be a couple of glycines or something, but it's essentially like a random assortment of amino acids that help create the perfect distance between these two things for the FRET to be active. And you can see that when they test these versions with the linker versus these versions which are shorter without the linker, they're getting higher fluorescence in all these four compared to all these that don't have the linker. So what they're doing with the linker is they're essentially trying to figure out like what is the best distance to have these things linked together to get a FRET, to get a good FRET combination. And then this thing is kind of key. And this was the hardest for me to decode and understand like what it was. So what is this pKa substrate? And why do they put this in here? And what is this testing? That's something that's like right here. Did anybody catch what that was? Or try to decode it?
How they actually bind it to the slide? Or
no. So this is actually the biosensor part. So this is where they're encoding the biosensor. Okay. And I actually had to look this up. So they cited the paper where this thing was developed, and I had to look up what this was. So I looked up this old paper. Here's the other paper. This is actually what that thing is. So that thing is a sequence of amino acids that holds the two sort of FRET fluorophores together in kind of like this linker region. And when this thing gets phosphorylated, there is a conformational change. So if it's phosphorylated, it'll conformationally change and it will bring the two fluorophores closer together. If it's not phosphorylated, it will be looser and they'll be stretched out far apart and the FRET will not be activated. So essentially what that little piece codes is it encodes a signal that if it's phosphorylated, it turns the FRET on. Okay. So this is the biosensor part because what they're actually checking is this construct, these fusion constructs will not fluoresce unless they've been phosphorylated. And so that's what they're checking. That's the biosensor. And this is a specific substrate that receives phosphorylation from some particular enzyme that people care about. Again, like some of these things I care about, some of these things I don't care about. I care more about like the construct design. This is sort of like a tool to measure phosphorylation in biosensor assays. So if you want to know, is phosphorylation happening, you can program this little unit in between your two FRET sensors. And then if phosphorylation happens, you'll see a signal and that will cause it to light up. Does that make sense? Okay, so that was the hardest thing for me to decode. But once you sort of like decode like what the pieces of these constructs are, then it sort of makes the analysis a little bit more simple. Okay, so here's then the actual data where they're measuring like the best fluorescences of these things. And what would you conclude from this? Like, what's the conclusion from this data here? Yeah, like this, the thing that they engineered to the max, the supercharged RFP, was essentially the best fluorescence. Although you could argue that it has a lot wider variation. So there might be situations where you don't like that variation, but in general, it's the mean of its fluorescence is much higher than all the other means of the other fluorescent FRET combinations. So essentially like this combination is the best combination that you want to use for FRET analysis. So essentially what they've done is they've bioengineered the best FRET combination for future FRET studies using this system. Okay, let's see, what's we're talking about? Yeah. Sometimes linkers are not necessarily encoding distance. So that's a good question. So like if we were to talk about, let me get some space here and like talk about linkers. So a linker is sort of like the idea of you have A and you have B, and in biotechnology or in molecular biology, oftentimes you want to link these things together, right? And you might create some kind of like a linker for various reasons. One is that perhaps it's regulating the distance. But you have to understand that these are three-dimensional, like it's not just like a string, right? So it's possible, like I don't know what distance that this linker is causing. It might be spreading it out, or it might actually be like tightening it up, depending on the structure of the linker. The other thing that sometimes you want in molecules, sometimes the linker is, if it's a bunch of glycines, which oftentimes linkers encode, essentially what you're coding is like a hinge that introduces more flexibility between A and B. So it might be that without the linker, there isn't actually a lot of flexibility between A and B to move around. And it might be that when you add the linker, you get more flexible like hinges in the structure. I wouldn't be able to know precisely without looking at A, what the linker precisely is, its amino acid sequence, and B, where the linker sequence came from. There's probably some citation that like developed the linker. So I'd have to look into that to be able to answer that completely. But does that help? Okay. Yeah. Yeah, I think that's what I was saying. I think that's a good, I'm not sure. It probably depends on the bioassay. Maybe because this one does have higher variance. Maybe you want something instead that's much more tighter, like this one. I don't know. It probably, again, I'm not a FRET expert. That would be a question for like a FRET expert. Is it better to have more variance with just a higher brightness? Maybe that's better than a tighter standard deviation with lower brightness. I don't know. It probably depends on the context. So that's a question that I cannot answer. Let's see here. Then essentially they did this next part that they did was essentially like the same type of thing with a different application. So the next figure was, okay, let's try the same thing. We have this FRET combinatorial construct that we like with the EGFP and the STAG RFP, and they're checking it as a different biosensor. So in this case, it's a cyclic AMP biosensor, which is like some kind of a modification. And if they put this sequence of amino acids between the two, this thing can get modified. And when it gets modified, there's a conformational change, which then activates the FRET. So this is essentially, it's checking it with a different biosensor. So the first biosensor they're checking is phosphorylation. The second biosensor that they're checking is a cyclic AMP modification, which occurs on this like intermediate thing. So essentially just like saying like, are there more applications for biosensors in this? And they're testing that. And they show that it will work with this other biosensor. But again, like I don't take my word essentially, because the biosensor stuff is like the people who are interested in that are trying to analyze like complicated biochemical pathways. And so if you were actually interested in those, like then you would do a deep dive into how those modifications are actually changing. But the concept is the same. Does that make sense? They're just like checking it in a different system. And then 3, again, like I don't want to hammer too much on this, but 3 is essentially, then they test it with multiplexing. So the idea is, again, if you're studying biochemical pathways, oftentimes there's an enzyme A that does something to a substrate that creates some output. And then there's an enzyme B that does something to that, which creates a different output. And then there's an enzyme C, which does something to that. And each stage along the way, it's getting like weird modifications and it's changing. And as they go through these biochemical pathways, sometimes scientists want to be able to assess like the progress of something through a biochemical pathway. And so the argument that they're making here is that you can essentially use multiplexing of these different FRET combinatorial constructs to assess at what stage things are moving through biochemical pathways. And so they built these 3 different constructs. Here, there's an RFP fused to another RFP with these linker. Here's a yellow Venus fused to the RFP, and here is a T sapphire, a blue protein fused to this RFP. And they test these all like in the same cellular system, which is multiplexing, to see whether or not they can see something transitioning through an actual like biochemical pathway. Again, like I'm not personally actually even interested in the ERT, the AKT, the SRC like pathways. That would be something if you're really interested in like deep dive biochemistry, you can look at those pathways. Essentially, what I care about is they're building different constructs to actually watch something visually transition from one stage to another stage to another stage, and they can actually quantify the transitions by the fluorescence of these fluorophores with the FRET assay. That's essentially like what I get out of this. Okay, final things to talk about. So one of the things that they that I think is worth pointing out in the discussion, let's see. So they try to surmise structurally like what might be happening here. And that one of their hypotheses is that if this is the RFP, and this is the thing that could go into the dark state when that one protein binds to it, they suspect that when that protein binds to this position, essentially they think that there is what they say here, and they read, if you read this, is they think that there's like a lever system. So here they say, acting as a charged lever to the internal environment. So what they're saying is when that other protein, what was it called? Drowns P or something? What? Drown. Drampa. When Drampa, it's like a weird dwarf name. when Drampa binds to this D159, they think that that creates like a leverage, like it's actually like pulling on this residue, and that is creating a lever that is essentially like levering out the beta barrel. So these beta barrels then are like getting, I don't want to say occluded, but like levered out to sort of like destroy the fluorophore. And that is what's causing the blinking, is what they suspect. Again, this is like, you would need intensive crystallography to actually prove that, but that's their hypothesis for what actually causes the dark state blinking. And so all you need to do is sort of disrupt that binding site. And if it can no longer bind at that spot, then the lever can no longer be activated. And so you don't anymore have the blinking. And that's the gist of the paper. Were there any questions or anything? Or I should ask also say, like, do you guys feel like when you were reading this, maybe you didn't catch everything, but were you able to understand most of the words? Or was it like, no, totally lost?
It was like not
what about, okay, so like, but what about, did anybody actually read the methods and be honest? Like, raise your hand if you actually like looked and read some of the methods. Skimmed them? Okay, so did you look at, actually, okay, so I did want to talk about, let's look at that quick because we have some time. So I did want to talk about like this paragraph in particular. So this is a paragraph in the methods, biosensor cloning, where they actually are talking about the process of how they built all those different like constructs. That's what we are trying to teach in this class. So my concern is, can you read this and can you understand like what's going on? So let's go through it together. So cloning and subcloning, like we all know what that is now, were performed using DH5 alpha strains. So we've talked about different bacterial strains. Here's a bacterial strain that they do their cloning in. Of E. coli. So they're using E. coli. We've taught all about E. coli. These are mammalian constructs, and they were cloned into the pcDNA 3.0 vector. So all these constructs, essentially, they're building it into a backbone, which is named pcDNA 3.0. And if we wanted to know like what that backbone was, we could look that up and we could discern what that was. So you should like, hopefully everybody could understand that, right? Maybe you didn't read it, but now you should be able to understand it. And then let's look and see. Modified multiple cloning site and constructs were cloned into this vector. So what method are they using to assemble these constructs? Well, this is I think this is the acceptor, but my main question is what we talked about many different ways to do like insertions into plasmids, insertions into chromosomes. What method are they using to insert the things that they want? Yes. So again, you should be able to read this and you should be able to see that they are using restriction enzyme cloning. So it says biosensor inserts were first subcloned into PR set B. So they cloned them into some vector using restriction enzyme cloning. So they first like captured them with restriction enzyme cloning, and they're using on the left BAMH1, on the right EcoR1. We know what those are. Those are restriction enzymes. The C-terminal fluorescent protein was then replaced by PCR amplified stag RFP. So they essentially like built one part of it and then they modified a second part of it using PCR restriction enzyme cloning. So they PCR amplified some sequence that they want to insert. And when they did that PCR, they built into their primers 5' SAC1, 3' ECO R1. So they're using multiple stepped restriction enzyme cloning. We should be able to at least understand that. What? Okay. Yeah.
We made that one GMP and one IP in the same
Yeah, so they're putting multiple. No. So if you look at, no, they should not have their own promoters and terminators. If you look at the construct, this is a fusion construct. So there is in all these constructs, the start codon. Maybe if I can do this. Perpetually cyber attack. All these constructs. This is the start codon, and this is the stop codon. So all these are fusion constructs, and the promoter would be up here. So these things, they're creating this. This is one of a fusion. I can't understand. Yeah, there's a single in these constructs, there's a single promoter up here, and there's a single terminator on this side. Yeah, the linkers you use in fusion constructs to generate some flexibility between the fused parts. Yeah. Okay. Let's see. Back to this thing. Again, they're just talking about more different restriction sites that you're using. And if you really like wanted to decode stepwise what they're doing step by step, you could read this very in detail. You should be able to understand like what's going on. Anything else we're talking? No. So after me sort of like pointing this paragraph out, like, do you kind of understand? You understand the methods of what they're doing? That's where I need you to be at. So you might not understand every single detail. You might not understand all the data. You might not understand all the biology of all the different biochemical pathways, the circ, ERK, whatever, all that crap. But you should be able to understand the biotechnology, which is, okay, they're building different constructs. How are they building the constructs? How are they engineering the protein? How are they selecting for various phenotypes that they want? And then how are they making mutations? How are they doing SDS page? All that stuff should now be like understood. Okay, so that's all I wanted to communicate by discussing this paper. That I'll end it there.